Hey, you guys. So we're going to talk about the Calvin cycle, the second part of photosynthesis. So remember that the Calvin cycle is fueled by the ATP and the NADPH that is created during the light reactions. And so the Calvin cycle is similar to the citric acid cycle in that it's a set of chemical reactions that are fueled by enzymes, and there are metabolites created at the end of each reaction, and that it's a cycle. And so the uh, the, the citric acid cycle in cell respiration used oxo, ox, oxoalacetate. Can never get that right. Um, it was produced in the last reaction, and it was used in the first reaction. And so the same is true here with RuBP. It is created in the last reaction and is used in the first. And so the difference here is that the citric acid cycle was catabolic. It was breaking down molecules to produce ATP. And here, this is anabolic. We are consuming energy to build up molecules or build up carbohydrates. And so it uses the ATP and the NADPH of the light reactions. And so glucose is not directly produced from the Calvin cycle. It is G3P. That is the goal of the Calvin cycle. And so it takes 3 G3P to make 1 glucose. And there are 3 steps in the Calvin cycle. We're going to call them phases. Phase 1, carbon fixation, and that is going to be right over here. Phase 2 over here is the reduction, and phase 3 is regeneration of RuBP right here. So let's go back to phase 1. Phase 1 is occurring in the stroma, and there are 3 carbon dioxides. So if we follow this process here, there are 3 carbon dioxides going into this process. So sometimes the book talks about going around in laps, and it'll take 3 laps. I don't want you to think about this like laps. I want you to think about it for 3 carbon dioxide molecules going into this process. There are going to be 3 5-carbon, what we call ribulose bisphosphate. But from here on out, we're going to call it RuBP, and that's what's going to be used in the last set of reactions. So RuBP is generated in the last step and used in the first step, just like oxaloacetic acid. The enzyme used to fuel this reaction is called Rubisco. And Rubisco is famous for the fact that it is thought to be the most abundant protein on Earth. It is the most abundant protein in the chloroplast, and there are many, many, many chloroplasts in each leaf. So there are many, many Rubisco molecules on the surface of the Earth. So phase 2, reduction. So we are over here. This is where we're generating our G3P. So our input of energy, ATP are used, NADPH are used to fuel this G3P, which is technically a sugar. And so 6 G3P are produced. 1 is a net gain of G3P. The other 5 go on to be converted into RuBP. So remember that it takes 3 carbon dioxide molecules to produce 1 G3P molecule, or a net gain of 3 G3P. So it takes 3 carbon dioxide to make 6 G3P, but only 1 net gain. The other 5 go into phase 3. And so remember that 3 CO2 per 1 G3P, and so therefore it would be 9 CO2 per 1 glucose. Phase 3, regeneration of RuBP. So we are over here. 3 ATP are used to rearrange the 5 G3P that went into the process. They are going to be used in the first step. They will take on carbon dioxide and go into the cycle again. Those are a set of steps that are far more basic than in the book, but this is the extent that you need to learn it. So the Calvin cycle on the whole, let's look at the final outcome. The Calvin cycle consumes about 9 ATP and 6 NADPH for each G3P molecule produced. And the G3P don't produce just glucose. They can be used to synthesize disaccharides or polysaccharides. And that is going to be another set of metabolic reactions to produce those molecules. Okay, have a great day.